queries, and then those can be resolved. So today we will be seeing all the basic things like basic techniques of open source molecular graphics programs and what is its significance. When you talk about this open source molecular graphics program, it requires a lot of softwares, okay, and you don't know when you start your research, you don't know which software to choose, and based on your seniors, based on your teachers advice, based on your research papers which you saw here and there, and you take one program and follow that program and go ahead, which is not a correct practice. So when you attend this type of program, you understand what is the potential of choosing a correct program. That is a very important thing which you should understand. You should follow that program and also have a comparative study, which is also required. So what I feel is that if you don't have a comparison, you think this is okay. So it is happening with most of researchers, including us. So we think this is good and we proceed with that program. We finish the work, we publish paper also, and we publish not in a very good journal. So our target when you attend this type of program as a researcher, as a teacher, as a scholar, you always think about a good journal. So for that, if you read this today, I will brief few of them. Not all is possible in 1 hour, but whatever best is possible, I will try to tell you. The motive behind this session is that you should know what are the programs, what are the significance. We are not recommending that this program is good, that program is good, this software is good, or that software is good. There are many students who have been registering for PhD or registered for PhD, some master's student, some faculty members, and some senior postdocs. So this bunch has all the experience. So the guidance is only for right type of software. One more thing before I start the lecture I want to tell you is that when you are choosing any program for graphics modeling, okay, or simulation, you should have your system ready with it, because many times the system requirements you don't check, because we are not computer science person, we are biology person, so we don't get it checked properly, the computer and systems require, which is very very important. Because what happens, it happens with my students also, it happens with your colleagues also. After some time your program goes slow down, work aborted, and many times you lose lot of data. It happened. So 4 days, 5 days, then you cry and think that okay, 4 days work is gone, because you have not taken care of the systems requirement. System requirement is very important. So those are the very general things which you need to know and note it down. I will also suggest you a better system requirement in a separate email or document, where you need to see how this system can be achieved. It is not possible to everybody to have that system, okay, so you can use the various type of other remote assess location softwares to assess that system, okay. Like if I don't have a system, I ask the computer science department, but they cannot have all the time open their department. So these are all practical things which before the lecture I will tell you. Now if you see this presentation, let me go through one by one. Okay, what is happening? ढीला डाला है क्या ये? This is, I didn't disturb anything. Because I think loose है थोड़ा, है ना? No, first it came and then some direct लगा दे, है ना? Adapter not required. If it is adapted to Apple, then we can keep it. Okay. Okay, so we carry on, meanwhile they fix. So when you try to see that what molecular models you are trying to use, what molecular model is required for your research, then it should have molecular modeling, it should not have molecular modeling. It is not mandatory that you use the software, graphic software, it has molecular modeling or not. Okay? And the third is the what macromolecules you are targeting. So either it is a DNA or it's RNA, okay, or it is a protein or different type of other complexes. Then what are the 3D view opportunities available? Okay, how 3D view opportunities are available and how you can see these opportunities as a research subject. Okay, so these are very important parameters when you use that. Now, representation of molecular models. So there can be various type of representations. Okay, so one is this atomic representation, which is very attractive. So when you talk about atomic representation, we don't see atoms at our lab. So being a biology student, we are always attracted towards chemistry. And when we are attracted towards chemistry, we need to see that ball stick model, stick and space filling model, and these representations of positions of atoms and covalent bonds which you cannot understand without these software. So this is one. Then backbones. This is also very good because when you work on long peptides, long proteins and other chain based molecules like also the monoclonal antibodies or something like that you are seeing, or immunoglobulin also you want to see, you have to see all those backbone structures. And also when you talk about backbone structures, you have these opportunities to understand various type of proteins, not only one type of protein, you can see many types of proteins, and the target protein where you are working template is also available, so you can compare your structure with that particular protein. And the complexes. The major challenge is understanding complexes. Many times we feel that we have understood the enzyme, we have understood the protein, RNA, everything, but the complexes which are very important. And I shared one paper with one of my postdoc, and we can see that most of the specific proteins, they are not yet fully discovered. Discovered means function. So maybe one function we know, structure we know, we don't know the second function, third function. So this is the novelty where you can have more opportunities for understanding that protein and complexes. Okay, then stability of the complex, which I will be taking up later on. But this is a very important statement, that the complexes, how to understand the complexes. Complexes, word is complex. So you see the complexes understanding is also a very tough job. So tough job but not impossible. Okay. So tough means possible job, but you need to put your efforts. And function also. Function I have not written here. Now examples of molecular database modeling databases. You know these databases, but I want to tell you few specific things about them. The PDB, 3D structure of protein, most proteins are available in the PDB, but there are few other proteins databases also, Swissprot and other, where you can get some specific protein. If you are working with specific organisms, they have a specific databases, like Cyanobase is there, okay, and those, like for cyanobacteria, you may have some other databases, local databases, where you have more information about that particular group of organisms. So it contains nucleic acids, complex assemblies, and if you see, this is a recent data that 1,70,000 structures are already defined, and you can use these structures for your research, whatever you want to do. The theme of the GAN workshop is for drug design. Okay, drug design doesn't mean that you go ahead with the drug development. It means understanding what type of drug is required, what type of drug is not available in the market, what type of drug should be targeted. So these are the important things which you can see, and if there are so many structures available and you have targeted one or two proteins, so it is easy for you to understand that. Then PubChem, this is also very common database, and which is used by many researchers, and it contains small molecules and bioassays. Like you know, bioassays are very important when you talk about drug discovery. So bioassays information you can get it, and it has more than over 100 million compounds, and it's a valuable resource for drug discovery and chemical biology research. Okay, chemical biology is another important branch. Okay, so being a biology person, if you know chemistry, if you know little physics, and if you know little other aspects of mathematics, you will be different from others. That is for sure. Okay, so we are always, I am very afraid with mathematical equations and chemistry part, it all confused me. Okay, so I always get understanding that if my student, if you people have more knowledge about chemistry and mathematics, that will be good for you. Okay, if you don't know about Professor Liang Tong, you may have Googled yesterday. If you see his biography, he was a chemistry student. Okay, and he studied mathematics, okay, physics. Okay, later on after BSc and BS program, he was interested in biology. So strong base of chemistry is required, and those students who have a good chemistry, I always sit with them and understand what they understand, and sometimes they describe me, sir aapko samajh mein nahi aayi hai, I will explain it. Okay, so this is a very important thing, and that's why here in this you can see that when you talk about this compounds, understanding compound is not a cup of tea for a biotechnology person. Even if you have database, sab kuch, you know everything, then also you don't know that okay, this compound is like this, the structure is like this. So these are the challenges with you, and the challenges can be recovered, can be achieved by understanding and collaborating. Now this is the third one, RCSB, Ligand Explorer. It is a web-based tool which allows users to search and visualize a small molecule ligand in the PDB. It provides detailed information on the ligand's structure, properties, interactions, and protein. This is also a very good software and database. Ligand Explorer, I don't know how many of you have tried it, but those who are working on protein modeling, they always look for it. Okay, so because you need to understand the ligand binding. Okay, why we are understanding all that, it is for the quick, before the emerging of bioinformatics, before the emerging of this subject computational biology, how the researchers were doing all these jobs. Like if you want to screen any enzyme, if you want to understand the efficacy of any enzyme, you need to do several reactions. Okay, now if you are working on an enzyme which is required for pulp and paper industry, or it is required for fruit industry, okay, how you select it? Based on your various screening experiment, primary screening, secondary screening, tertiary screening, and then you understand, okay, this enzyme is binding with chitin, it is binding with xylan, it is binding with cellulase, cellulosomes, and all those interactions you see, and then you decide. With these softwares, now you know what enzymes, what ligands, and you finalize in between. So it is called pre-screening. So with the help of bioinformatics, you can pre-screen which enzyme can be targeted and what type of ligand to be selected, what active site is present, why this enzyme is not active for this type of purpose. So many things you do with the help of bioinformatics in quick time. So we screen 5 enzymes and we try to see the applications. And when you understand the application, by the time you complete your 3-year, 4-year, with the help of bioinformatics, with the help of these type of softwares, you understand the ligand and biomolecular binding together. And you understand that, okay, this enzyme is able to bind, this is the active site. If we have some changes in the active site through molecular modeling and we see there the wet lab results, then this enzyme can be more effectual. Okay, so and a normal enzyme and an enzyme which is engineered enzyme, what is that? This is the purpose of engineered enzyme. So when you do, and it is not possible that only the bioinformatics and the software will do the job. You have to engineer the enzyme, you have to see the enzyme with the wild type enzyme, what is the natural enzyme, and then try to see the compatibility whether the engineered enzyme is better or not. So this is the ChemSpider software, another one, okay, and it has most of the chemists, biochemists, if you see during the COVID time, drug discovery, thus bioinformatics helped a lot, because most of the time people were doing the modeling work, they were doing the simulation work, they were seeing what is the binding pattern, what is the immunogenicity, everything, okay. And I will share a paper with you which we wrote during COVID also, that how bioinformatics, structural biology techniques, advanced computational biology techniques help to develop the vaccines, especially the epitope design. Epitope design is also a very good, interesting area, and that is very important to understand that, okay, these are the softwares which you make you understand that, okay, you can do the work in silico, then do the work in wet lab. So this ChemSpider is also one of them, and based on that, the information to be collected, and also you can screen the priority list based on your requirement. So this is there. Then how can we access to molecular modeling database software? So this is a very simple question, not a complex question. So web-based interfaces are there. You have the web-based interfaces, and here you can, most softwares are freely available. Okay, so you can use those softwares. Which are not available, if you put your email ID, they send you the license. Sometimes they ask for the academic ID. Okay, this is a big question. And sometimes students, they ask that we don't have academic ID like ac.in, so we don't get it. Institute ID, right? So for that you can use the ID of your mentor whom you are working. If you are a MSc student, sometime you get ID, sometime you don't get. Okay, but as a PhD student you always get. As a postdoc you always get. Okay. So for MSc, I know that most of the master's students, they don't have time. They are always busy, so they don't get understanding of these things. They just read in the class and then marks mile khatam. So this is all about MSc. But in PhD, you always get an academic ID, and after academic ID, you can download these softwares. You can use this software. But one thing which I want to tell you, because it's a mandate of GYAN program, that ethics is very important. So whenever you use any software from any university or any databases, please mention it in your acknowledgement or provide the link with proper details. Okay, because these software are open source software, so they are made for you, so that if I can use it, you can also use it, or other person can also use it. But while completion of the experiment, after completion of the experiment, please acknowledge that thing. So this is one thing. And then software tools. This is many molecular modeling databases provide software tools that allow users to assess the database and perform computational analysis and simulations. These tools are free to download, and they can be used, but some may require a license or subscription, which I have already told you. Subscription is costly. Okay, so I will not advise you for subscription. Okay, if you want any help, you can always connect with fellow researchers here. They are all well trained. Okay, many of the postdoc teachers who are sitting here, they know how to use that, and you can get always the subscription based also support. Okay. Now, application programming interface. This is also a challenge. So few days back, where is there? It is not for the molecular modeling, but MATLAB is there, which is used for various type of process optimization. You can use the softwares which are used for molecular modeling and graphics. So if you see the list, we will provide you the list and the links to download. I think one annexer is there, but we will provide you that. And when you talk about this programming interfaces, these are to be trained properly and they are integrated together. So if you don't download the whole package, you don't get the features. So sometime it happens, software chal nahi raha hai, theek nahi chal raha hai, it is not happening, okay, and it is not properly installing. It's a simple thing. You have to have the all package, all the files together, and then you know how to install the software. It is also not easy, okay. So I am also not trained properly, so I also learn. So same way like that, we have to understand how to install it. It is not like simple other softwares, okay, which you download and install, and okay. It is not like that. There are some specific type of links available there, and also there is a flow, workflow is there, okay. So you need to understand that. So please note it down, that software package, full package, and its annexes, very important to understand all the features of the software. Otherwise you will have a limited software or your program will not run. So you will again complain, sir told me that it will work, but it is not working, and it is not working even after trying multiple times. So you need to understand. Otherwise there is a web support, you can send the message to them, they can also help you, okay. Then data download. Some molecular modeling databases provide downloadable data files that contain molecular structure and related data. These files can be used with molecular visualization software and other computational tools. So this is also a very general statement, but downloading also has its limitation. How many downloads you can do per day, and what is the size of download, where to store the download, how to open the download. The biggest challenge that sometimes you open the files and you are not knowing what I have opened, what I want to see, okay, and you are I am telling you a very basic problem which a biology researcher face, okay. And also sometimes you don't have a supported file system to open the file, okay. So like if you have downloaded some SVG file, you don't have SVG software, you can never get all those details of the structure. So you have to have those type of extensions available. Then features of molecular modeling database. This is with all the molecular modeling databases, most of them, okay. The first is structure search. You can do the structure search, and also using a range of criteria such as molecular weight, functional group, substructure search, 3D visualization, how you can use the 3D structures and interactions between molecules, then chemical properties, to retrieve a range of chemical properties like boiling point, melting point, solubility, other parameters. Then biological activity, the ability to retrieve information on biological activity of molecules such as enzyme inhibition receptors, binding and toxicity. So this is also very important parameter. Biological activity, chemical structure, 3D visualization and structure search. These all things are available and available in the molecular modeling databases. Now applications are unlimited, okay. I am highlighting few of them, but they are just examples, okay. These unlimited applications. The first is the protein engineering. As the word suggests, engineering, okay. It means you should have the capability of engineering something, okay. So you should have a strong background, and you see you can use these databases to design and optimize protein with specific properties such as enzyme activity, binding affinity and stability. Enzyme, when you work on enzyme, it is a very challenging area, okay. And if you are not able to get a proper enzyme characterized, all these softwares, databases are of no use. So you need the structure, proper details and specific characteristic of that enzymes. So when you talk about protein engineering and you are targeting that, okay, I will have a protein which or enzyme which has 100 IU activity and it is having a very good stability at 65-degree temperature. So how to design that protein? You need to understand by seeing what type of ligand, what type of interaction, what type of substrates, and at what time the protein is denatured or degraded, and why this protein is not denatured even at 60. If you understand that, like there are thermophilic proteins, thermophilic enzymes which are not degraded at 50, 60, okay. So you need to understand that, and some proteins, some enzymes, they degrade at 35, 30 or maybe like that. So you have to understand that, which is possible with the help of this softwares. And also researchers can use the database to search for protein structure and similar folds and functional sites, then use the predictive modeling tools to engineer new proteins with improved properties. This is in silico. You can engineer protein in silico. You can generate a protein, new protein, okay, structural details of a new protein, and then you can also understand that this protein is a new or engineered protein. But whether it is viable or not, it is all through wet lab exercise. So you have to do that wet lab exercise to understand that. But with databases, with other things, you can do the protein engineering research. Then drug discovery. This Professor Tong is already covering. I will not throw much light on it, but this is recent days demand. Okay, drug discovery. Okay, how many people they are trying to get the new drugs. Okay, novel drugs, novel medicines. Okay, and now there are some more specific agencies looking for researchers who can help them to understand the whole personalized medicine domain. Personalized medicine, because that is coming up area. So they want to know the databases, they want to know the genome of person, they want to see which type of drugs, which type of medicines are to be given to which type of individual. So this is a very, very interesting and challenging area. So if you think about it, personalized medicine, this drug discovery and molecular databases, they are very helpful. Somebody wrote to me a few days back, one of my previous lab mate, he is in Australia. So he wrote to me and he told that, sir, I want the databases of the cancer patients, their genome and their, because I want to know if the 1000 people are treated for the cancer in the IMS, I want their data, what medicine they have been given, what efficacy they got. In the same way for tuberculosis, all the diseases, okay. So they want to know, and the functions, structure and function both, okay. So this is a very important thing. And drug discovery, if you see here, specific chemical properties and biological activities that can use the predictive modeling and also see the potency, okay, which is very important, potency of the specific medicines, okay. Medicines is active, medicine is not active, medicine is effective, medicine is giving good effects, medicine is curing the disease, medicine is controlling the disease. All these aspects comes under potency, okay. And this potency is very important when you talk about drug discovery, okay. Any drug developed by any of the companies, if it is not a potent drug, it will not be available in the market for the longer time. So this is the biggest challenge, okay. In case study, I will cover this drug development little bit with wet lab and dry lab and how metabolic engineering can be used. And we did one collaborative project in 2013 to 16, probably 17, and it was with IPCA Industries, where one of our PhD student, and now he is the general manager there. So we tried to develop a new map, okay. So that was based on in silico and wet lab experiment, and along with the engineering, metabolic engineering. So all these tools, they led to a new drug development. And if you develop a new drug which is really good and potent, it will be always in demand for the industry. Now, one thing is there which I cannot deal much about, that is the pharmacokinetic properties. Okay, it is a special domain where pharmacokinetics properties are analyzed, okay, where you also know that how the drug will be effective in the plasma or serum, whatever they have. So this is another domain, okay, you can read it whenever you have time. Now, biophysics, okay, this is one area which is required, and most of the students who are studying their master's course, or they have studied their master course in biotechnology or in microbiology, they have not read this biophysics, okay, but this is important. So when you choose the course for your SWAYAM or something, please read this biophysics also, okay, because at least you can have additional course. So biophysics will give you the basis to understand all these molecules. Then enzyme engineering, this is a new discipline, and many of the researchers are working. So in drug discovery, you also understand the enzyme engineering aspects. I will cover this, but enzyme engineering may use the recombinant DNA technology, but you should also see that whether you are really looking for engineered enzyme or not. Okay, when you have a native enzyme which is doing the job, so engineered enzyme is not always required. Engineered enzyme for many other purpose. If you see this photo, okay, it's a simple photo where we wrote a review in 2020, okay, and I thought we can work on that, but unfortunately not much we have done. Okay, so if you see this photo which is taken from our own paper, and I will share that copy of the paper also. Here you see it's a new area, okay, people are working on this area, and here also some students, some researchers like Dr. Pakumba is there, he is working on this area, microbial communities understanding, trying to see how these communities they affect the bioremediation capability. So this is one area where you use these tools, okay, genomics, transcriptomics, proteomics and metabolomics all together to understand and also understand that how the synthetic microbial consortium can be made, and when you are able to make this consortium, it will be definitely better than the existing microbial population or individual microbial population. Because when you work on consortia, like this is a group, okay, so you know संगठन में शक्ति है, okay, so when you work on that, it means that संगठन भी good लोगों का, ठीक है, it is not that you take people and they are trying to pull each other, okay, so that will not be having shakti, okay, so you should have microbes with variable capability, and you have to sort. It is not easy task, he knows the challenge, okay, and he has done the sorting, this microbe, that microbe, then try to do. And when you develop this type of strategy for your own research, it is only not for bioremediation, you can also see for other aspects, okay, like if you are trying to see the plastic degradation, if you want to see the oil spills, you want to see other aspects, okay, polluted sites remediation, so this is very important. So this is a snapshot where you understand that, okay, you use the tools of systems biology and try to develop a synthetic microbial consortia which will give you a better effect for environmental applications. Then if you see this slide, this is also giving you the various softwares, databases which are used, and understand this concept in a better way, okay. So you see it is not only for drug discovery, it is for other aspects also, okay. So like in drug discovery also one thing is coming, ARG, okay, you know about ARG, AMR, antimicrobial resistance, that is also global threat, okay. So AMR tackling and drug discovery, okay, AMR is coming because of excessive drug medicines use, okay. So one hand we are talking about drug discovery, in one hand we are using so much antibiotics, okay, so we have to tackle this as issue as well. So this becomes again a challenge for us, that you have to see that the sites which are polluted with lot of antibiotic residues, the water bodies which are polluted with lot of antibiotic residues, how to remediate that. For that, this is the scaffold. You can also see that you can also do the pathway reconstruction, means the whole process is given here in this picture, and these are few databases which are applied for this type of research, and these are few databases, you can note it down if somebody is interested in this antibiotic pollution management or any type of bioremediation capability. These databases will be very important, and I don't know how many of you have read, gone through these databases. I know that like you see OxD base, this is first database used for detection of oxygenase enzyme involved in bioremediation purpose. Then you have AromaDec, which is online resource tool used to target aerobic degradation of aromatic compounds based on phylogenomic approach. MetaRouter, it is a prediction tool which provides information about pathways having meshwork of genes, enzymes that come from a diverging group of bacteria. BNice, okay, it is well-known computational framework that can deal with the metabolic pathways involved in the degradation pathway. It provides the framework to identify every possible chemical compound produced by the enzymatic biochemical reactions. Then RemeDB, it is recently developed prediction tool to study enzymes involved in bioremediation and metagenomic data. So these are the 5 softwares, tools, you can use it. And MSc students, those who are participating, or PhD students, if they work on any of the parameters, okay, and if you want to work on any parameter like pathways, okay, if you want to understand pathways, you can use it. Then you see, if you want to take picture, you can take it, but I think it is available in my paper, so I can provide you, and here also it is available. But this is good. I made this article in 2020, but I think very few students in my lab have used it. So you understand, Chirag Tale Adhera. Okay? So this is the biggest problem, that we explore new things, we have the capability, but we forget. So it is a human nature. Okay? So what happened? Even here the students who are working on various aspects, they can also use, and these are all free softwares. No charges there. Okay? Accessibility is also there. Okay? Those who will use, they will have a good data analysis. Those who will not use, they will try to struggle for understanding the data. And the problem with the software only is the understanding. After that, everything is clear. Like, it is simple like going in a road which is not suggested by Google. Okay, if you don't know normally we follow Google and we go as per Google. If you know any other route but you have not gone there and Google is not suggesting, then you will be just little bit worried about it. Kahan jayenge? How will we reach? In the same way, software. Once you start using the softwares, I guarantee you will like it, you will understand everything. And one thing I always tell my student, and I tell to me also, the best part working with computational biology is that the computer will never slap you. Is it happened? No. Kuch bhi karo, whatever you want, what command you give, he will never slap you. Why you have given that? It will shut down and everything, okay? But this computer will not be angry with you, and you will not have any type of problems with this. Okay, so whatever you want to do, you do with the computer. In the same way happens with when you work with microbes. Okay, there is no legal complication, there is no ethical issue. Whatever you want to do with microbes. Last 2 weeks we are trying with 2 different cyanobacteria, and we are doing lot of experiment with them. They have never said anything to us. Okay, sometime we change the parameters, we change the software, we change the process parameter, we change the strain, we change the conditions, everything our researchers are doing. And our microbes, they are telling nothing. They are doing the job. Okay, they are giving the signal that okay, this is not good. Okay, I will not produce more metabolites. This signal is coming. So yesterday also the evening I got the signal, and the result they told. So this is all simple things which you can think about and plan. So this software you have to handle. Okay, try to handle yourself. Don't think that I will tell you in lecture or explain you, and if you ask question I can resolve it. It will not be solved. You have to handle it yourself. Okay, like you do while you scroll on reels. Okay, same way you scroll the softwares. Okay, so I am not concerned about reels, but I am concerned about software today. That you scroll on software, try to see what new content is coming, what are different aspects available, what are new options available, what are the new applications available, and sometimes there is application notes also available in the software. So you can drag it and go to the application notes, see the related papers also. So this is one thing. Another interesting picture, okay, when you target novel enzyme for drug discovery. If you want to have a good enzyme for drug discovery, you can use this methodology and try to see, okay. And I have given example for this pollutant, but it is also common with the drug discovery. Getting good enzyme is very important. This is one review which I was talking about. It is very well cited. Synthetic microbial communities for effectual bioremediation. Here we have listed all these softwares, details, okay, and this paper is available for you. So you can get it from Shrabni. She is compiling all that. and this paper, full paper is available. Otherwise, it is on charge basis, so you can get it today here. And this paper will give you other ideas, where because we are always not interested in having the engineering of the microbiome. Okay, so that is also a very good aspect. Now, this is a key for today's lecture. Okay, key slide. Okay, and this slide again we made in 2010. No, not 2010, 2012 and published in 2014. Okay, so this slide I contacted this Nilsson and Otero. Okay, he made a diagram. Okay, and he kept the gap of this one. Okay, and this one. Okay, so what happened? The software where we are writing these models and this kinetic model. So he left with that. So I asked him that if you are working on this systems biology tools, why not we involve the specific models and also the kinetic models? Why not we integrate that to make an industrial system biology? When we talk about drug development, we should understand all the aspects together. It is not only that the drug development program will involve one step and you get a magic drug. Okay, it is not like that. So you have to target about industrial system biology, industrial approach, where you involve the tools of genome biology, you involve the tools of system biology, you involve the tools of chemical engineering, you involve the tools of metabolic engineering, you involve the tools of process engineering. So then only, and also the computational biology. So then only, if you want to work on any specific type of drug development, you have to think about this model and try to understand that how to get a high valuable productive strain of any specific type of clone or any type of microbe. So when you talk about clone development, I will take it in the tutorial part because उसमें कुछ experimental मुझे दिखाना है. Okay, so that is there. Industrial production host is there. You see, high throughput sequencing technology. So how to get a high valued clone? Because if you don't have a good clone, it will not give you a good profile of the product. So when you talk about the metabolic engineering, you should have high value, high production clone, and then also after you are getting all this data available, physiological experiment, transcriptome, metabolome and flux, you have to study. Okay, flux is also a very new field. We are not covering here, but we'll tell you in the tutorial little bit, not much. Okay, so flux balance analysis, FBA, also a very good thing, and it can innovate your data, which you have existing data. It can innovate your data and also try to see the targets of metabolic engineering. And then the kinetic modeling. Kinetic modeling is very important. Any drug development, I told you about the pharmacokinetics. I am talking about simple kinetics. Okay, whatever enzyme you are using for drug discovery, whatever metabolite you are using, whatever other metabolites you are working, kinetic is very important. Kinetic parameters are very important. So this slide gives you a industrial system biology, and it is a dynamic interaction between various disciplines and approaches. And we got this paper, I will show you, which was published in Briefings in Functional Genomics, Oxford University Press. I will show you that later on. Okay, this is also one slide which gives you protein prophecy for drug development. What type of protein you are looking for, okay, what activity. This is not very clear, but we have a small book series of system biology where we publish this whole for one microbe, one fungus, one enzyme, xylanase. This can be done with many other enzymes which are used in drug discovery. So it is not possible for one person to do that. It takes 1 hour, 1 year, 2 years, 3 years to understand that. So what is happening is that when you get the active site, or you are trying to see, you have to see that which computational function, what computational databases, what is the purpose of that protein, and how much is in the merit that, okay, if we change this active site, what will happen? if the protein is having more capability or it will be better for this type of application. So this is protein function prophecy, which is called as protein function prophecy. So this is another one, okay, which I want to tell you, still I have not explored it, okay. So all things I have not explored you, I am opening my ideas to you. If you want to get, it's open source, okay. And this is one where you can use, because cyanopeptides, I talk about it many times. If you Google it, cyanopeptides, how many peptides in the market, drug discovery, peptides in the market produced by cyanopeptide, you can get very few. So if you work, a cyanopeptide means they are generated from cyanobacteria. It is not means chemical based, okay. So you know the craze for herbal drugs. You see the Patanjali, okay, how much they have grown. So cyanopeptides means cyanobacteria produced metabolites which can be used as a peptide. So we have written one more write up there, you can read it when you get time. But you see this one, this is the PSP A1, this is something else, but regulation of this particular protein has not been studied properly, okay, because of information which are not much available, and especially with all the cyanobacteria it has not been done. All microalgae, maybe one or two, which is more extensively studied. So it happens that if one strain people are working, they will do working with that strain because everybody knows about it, okay. So as Google, I told you, if you don't know the road, you follow the Google, okay. So the same way you try to see that, okay, this is one, I have written this a very catchy title, tightening the screws on PSP in cyanobacteria, okay. I wrote some other title, but because it was invited article, the editor, she told me that I want to have a change in your article. I told what change? She told because you are really tightening various type of protein screws in your article, it should be like that. So the article was suggested, means the title was suggested by the editor and we did it. And if you see, I'm not going detail here because it is not all connected, but yesterday in the evening we had a discussion with Professor Tom when he was discussing about sigma factors. So then he told that sigma factors crucially, very crucial, especially for drug discovery also. But he has not thought much about it because he is already active in so many other fields, okay. So then this is the emerging tools in omics, okay. So cyanobacterial omics, because we focus more on cyanobacteria, single cell transcriptome, and single cell transcriptome is also a very good area, emerging area to explore. And now this is one case study which I am presenting. I will be covering more details probably in the tutorial. This is the HMG-CR, okay. This is the enzyme which is used for statin therapy. Statin, you know, कितने लोगों को statin पता है? How many of you know? 1, 2, okay. Statin is used for, बताइए? Leukemia? Lipidemia, okay. Statin therapy normally, in layman's language, because we are not doctors, we use it for blood pressure regulation, okay, with the help of hypertension patients, okay. So statin therapy is going on, still going on, okay. But the mechanism of action, how statin therapy is happening and how a better statin can be proposed based on the understanding, we started working on this, and it is all in silico work, okay. And if you see, this is paroxysmal HMG-CR, and how this enzyme is active, okay, and why a statin therapy can be should be improved, okay, because people are trying various type of statins every day, okay. So if you see this, we did the binding of HMG-CR isoform 2 with various statins through Discovery Studio. You can see that we have this pravastatin, cymostatin, lovastatin, all statins we binded with this, and isoform 2 was used, and then we try to understand that how this isoform 2 can have this statins connected, and how the statin therapy can be improved. Because existing statin, what is the binding pattern, how these statins can be improved. So when we publish this paper, you can read this paper full. I am not going detail because of the limited time we have. Okay. So when we wrote this paper, and this is 2012 paper, you can see here. Okay. Abhi 2025 chal raha hai. Okay. Still people are citing this paper. They are writing to us, and still there is nobody who is working statin and the isoform together, as well as their other parameters like kinetic, pharmacokinetic, all these things. Because nobody questions on that therapy. It is very successful. People, they are using that. So what is the improvement capability? It is given in this paper. And now this paper is, journal is very tough. I am also telling you. Computers in biology and medicine. If you Google it, you see the impact factor maybe 7 or 8, but rejection rate is 95%. Okay, means if you 100 papers are submitted, 95 rejected. Okay, without review. Okay, so they write thank you. Okay, thank you भी लिखने हैं बहुत अच्छा है. ठीक है? If they write thank you, you feel blessed. Okay, at least they wrote thank you. Okay, sometimes. Okay, so automated mail comes, thank you comes. So this journal is publishing this type of work. Why I am telling you? Because strong connection with biology, medicine and computer, this journal. So if you learn whatever in your research and it is connected to drug discovery, this is a good choice of journal. And also this study here, we try to see that how that isoform 2, which is showing less effectiveness, with HMGC isoform 1, okay, and we have taken these isoform from Homo sapiens. Okay, we have not done any mouse or something. See, one thing which I want to tell you very clearly, I am not an animal biotechnology person, but I fail to understand that if you do something specific to human, if you develop some drug with human, you have to have human trials. If you don't have human trials, your drug development is at stake sometime. No FDA, no drug authority will approve your drugs until your human trials are complete. So whatever experiment you do in mice, you do in rabbit, whatever you do, okay? And I'm yet to understand that why when these type of drug trials are given, it takes a lot of time. So the time taken also kills your all waiting time. So if you are really working on drug development program, devote 10 years, 15 years, 20 years for that, and then you try to understand that. And in the tutorial I will cover few of them, few slides given to me by Professor Tong also. I will try to explain you those slides, and I'm also reading those slides because very good research they have done at Columbia University, and I really admire their team, and they are really very focused. And if you see their capability of producing high quality papers. Okay, yesterday I was asking him how many nature paper you have. He told I don't remember. Okay, so you may have 15, 20 nature papers and science paper. Okay, natural structural biology. So this is all why he is here. We should also ask him that how to get this type of high impact papers and how to learn more things towards that. So targeting a new drug development is always a challenge. I have shown you few of the ideas and other discipline also, because most of you might be interested in the techniques part. Okay, so we will carry this further in the tutorial session. So I think this is over. If you have any questions, we can take up, because it's 11 now, and so this is acknowledgement, very important. Okay, so whatever I showed, Gyan, because of Gyan we are here. Okay, Gyan knowledge. Okay, global initiative for academic network. Okay, and I think this is the third Gyan program I am conducting. Two I conducted earlier in Marshid. I like this program because you get connected. One expert is coming, You don't worry about their various other expenses because covered by ministry, and he is here for 1 week. That is the best part. You develop the networking. Okay, like Dr. Mandeep is sitting. Mandeep developed a network with a researcher who came from Australia. Okay, and he is having, and China also. Okay, so they are connected. You get connected with that particular person. So it is not that you always get opportunity to go in that lab, but at least you can ask his opinion. You can get his help for various other applications or learning, and it's a mutual learning, collaborative learning. I have not shown that. Maybe network I will show you in the tutorial. Okay, how networking is important I will show you, and also tell you networking for understanding and helping. It is not one way. See, I will help her and she will not help me. Okay, it is two way. You have to help both ways. It is not that one person will help, other person will not help. So normally it happens. As a student, you can always help in knowledge gaining, understanding, okay, knowledge sharing, and with the fellow researchers. So acknowledge to IOE incentive grant which we are getting. Our School of Biotechnology, served DST funded for our cyanobacteria project. Our my previous university where I was for 10 years. Then this IARI, the cyanobacteria work, I collaborate with Professor Puppy, and then DST, Indo-US Science Technology, and American Society of Microbiology. It is a very good funder, especially for the young students. You join ASM, very good network, and very good people are there, especially at all levels, undergraduate and all that. Okay, and this American Society of Microbiology is connected with other societies, so that is also a very good network. You can join as a student member. Sometimes they offer a lot of discount. Okay, and you can join them. If you don't join them, visit website, see article, read it. Okay, there is no need to join also. Okay, and if you ask, I can send you my newsletters, my information to you. Okay, then University of Cincinnati, USA. Okay, so thank you very much. If you have any questions, you ask it, but not the tough question. Okay.